Dear students, we request all of you to join the meeting with your register number followed by your name. Students, kindly rename your identity with the register number and your name.
மேடம் ஆடியோ வந்து மேடம் ஆடியோ சரியா கேட்க மாட்டேங்குது மேடம்.
சார் இப்ப கேக்குதாங்க சார்?
இப்ப கேக்குது மேடம். கொஞ்சம் சவுண்டா இது பண்ணிக்கோங்க, இல்ல மைக்கை பக்கத்துல வச்சுங்க.
ஓகேங்க சார். ஓகே. Dear students, we will start the program by 2 minutes. Please kindly ask your friends if they have not joined to join fast. We are about to start the program.
Hello, ma'am. Oh.
Good afternoon, students. We are happy to welcome you all for our inaugural function. And now I request Sai Shweta to start the program.
A very warm and healthy afternoon to all dignitaries, guests, and delegates with great joy and immense exultation. Great things happen when there is a gathering of great minds. It's our privilege and pleasure to extend our welcome to all present here for this auspicious inauguration of Creauctus Association activities 2020 to 2021. Now, I'd like to call upon Ms. S. Vijayalakshmi of 2nd BSc Biotechnology in the prayer start.
கலைகள் வாழும் இடமெல்லாம் இந்த மகள் நலம் புரிகிறாள், கல்வி அமுதம் அருள்கிறாள், சொல்லி சொல்லி தருகிறாள். திருமகள் இவள் கருணை தானம் அறிவு தாகம் தீர்க்குது, செல்வம் கல்வி இரண்டையும் நம் வாழ்வில் ஒன்றாய் சேர்க்குது. வித்யாலக்ஷ்மி வருக என்று வீணை நீட்டி பாடுவோம், நித்தமும் அவள் புகழை போற்றி நர்த்தனங்கள் ஆடுவோம். கலைகள் வாழும் இடமெல்லாம் இந்த அலைமகள் நலம் புரிகிறாள், அலைமகள் நலம் புரிகிறாள்.
Thank you, Vijayalakshmi. Greeting. the universal tradition that ought to bring profuse joy and pleasure to both givers and receivers. I'd like to call upon Ms. K. Nandita of 3rd BSc Biotechnology to welcome the gathering.
A pleasant good afternoon to one and all. I hope all are safe staying home. I am very happy to welcome you all to the inauguration of Creauctus Association activities academic year 2020 to 2021. We all bow our head to the holiness of God who showered us with blessings. I am glad to welcome Thiru K. Palanisamy Avargal, the correspondent, Congo Arts and Science College, who supports in all our activities and upliftment of the institution. We welcome you, sir. I am very happy to welcome Dr. N. Raman, sir, the principal, Congo Arts and Science College, who is the pillar for the development and welfare of our institution. We welcome you, sir. Next, I am very excited to welcome our special guest, Dr. Prashanth Kumar Kandalla, sir, staff scientist and senior postdoc, stem cell and macrophage biology lab, Center for Regenerative Therapies, Dresden, CRTD, Technical University Dresden, TUD at Germany. We welcome you, sir. I am very grateful to welcome Dr. C. Deepa ma'am, Head of Department Biotechnology, enriching our academics through her tireless efforts. We welcome you, ma'am. I am very happy to welcome all the heads and staffs from other departments. I welcome all the teaching and non-teaching staffs from Department of Biotechnology, and I finally welcome all the students of biotechnology. Thank you.
Thank you, Nandita, for your humble and cheerful words. Strong roots produce beautiful flowers by supporting the trees strongly. So does our principal, sir, supporting our institution and producing intellectual graduates. I request our correspondent, Sir Thiru K. Palaniswamy Avargal, to present the presidential address.
Sir,
Ma'am,
come
good
on.
afternoon and all. I am pleasure to introduce our chief guest to you, a new scientist from long ago, and I congratulate and wish all the success for this department growth through our HOD Deepa ma'am, and I also congratulate all our faculty and teaching staff, non-teaching staff, lab assistants for supporting for the growth of this department. And I am very much thankful to all the students for giving participating for this program and giving a leading success for this, achieving in each and every goals for the supporting for the head and the lectures. I once again thank all of you, and I thank very much for our principal sir, Raman sir, for giving a very big hand for your supportive to this department, particularly for this department. He supports very much to give and bring it to a first position in the South Zone. I thank you all the participants, and I welcome our special chief guest for this program to continue the program to grand success. Thank you.
Thank you, sir. Thank you for your kind words, sir.
Thank you, sir. I'd like to take this opportunity to invite the one who has been our guiding light and has always encouraged us to give the best. He makes the task of running this institution look so smooth as he effortlessly manages this mammoth task. I request our Honorable Principal, Dr. N. Raman sir, to felicitate the gathering.
Dear students, due to work in our college, sir was not able to join, but he has conveyed all his wishes and blessings for our department activities and for our department growth. So we will continue with the next. Sai Shweta.
Sure, ma'am. Now, I'd like to invite Ms. T. Swaker of 3rd BSc Biotechnology, Secretary of Creauctus Association 2019 to 2020, to enlist the activities of previous academic year.
Good afternoon, everyone. Annual report for the academic year 2019 to 2020. The Department of Biotechnology was recognized and funded under DBT Star College scheme, and all the activities of the department were carried out under DBT fund. The Department Association inauguration, Creauctus 2019, and the guest lecture on plant biotechnology was held on 29/6/2019, and the special lecture was given by Dr. P. Parameshwari Ma'am, Senior Scientist, ICAR, New Delhi. The following activities were conducted for third year students. Hands-on training program on mushroom cultivation was conducted in the month of August. An industrial visit was arranged for the third year students to Parle Biscuits Limited, Chennai in the month of August. And in the month of January, a workshop on animal handling was conducted. In the month of February, third year students were taken to Pasteur Institute of India, Kunnur as institutional visit. The following activities were conducted for second year students. A guest lecture on application of mathematics in biology was organized in the month of August. An industrial visit was arranged for the second year students to Central Silk Board, Mysore in the month of August. And in the December 2nd, biotechnology students were taken to Common Effluent Treatment Plant, SIPCOT, Perundurai as a farm visit. A guest lecture on career opportunities in medical transcription was arranged in the month of February. The following activities were conducted for first year students. An induction program for first BSc Biotechnology students was held in June month. Guest lecture during the celebration of International Year of Periodic Table for first BSc Biochemistry, Biotechnology and Physics students was conducted in the month of August. An industrial visit was arranged to Central Leather Research Institute, CLRA and Birla Planetarium in the month of November. Hands-on training on histopathology and chromatography was conducted in January. In the month of January, first year students were taken to farm visit to Arvind Dairies, Chittor. In the month of January, a guest lecture on emerging trends in computational chemistry was organized for first UG students of Biochemistry, Biotechnology and Physics department. A workshop on bioinstrumentation was arranged. Hands-on training on hematology was arranged in the month of February. The following activities were conducted to UG Biotechnology students. Competitions related to periodic table for DBT Star College department students had conducted on 13/8/2019. Science Fest, an inter-school science exhibition contest for school students, was organized by DBT departments. The DBT departments of our college, in association with Tamil Nadu Science Forum, hosted district level 27th National Children's Science Congress 2019. School students from all over the districts presented their projects on the focal theme Science, Technology and Innovation for Green and Healthy Nation. A guest lecture on importance of ISO auditing in food industries and career opportunities in ISO auditing was arranged for the UG students of biotechnology. A motivational talk was planned and executed in the month of December by DBT Star College departments on the topic Focus, Learn, Grow and Enjoy. In the month of January, the Health Club, a part of Creauctus Association, organized BioFair, an exhibition of natural and value-added products. In the month of February, the department organized Insta Crackers, an intramural quiz competition for UG Biotechnology students. An outreach program for self-help group on mushroom cultivation was organized in the month of February. In the month of February, National Symposium on Mitochondria in Health and Disease was jointly organized by Society for Mitochondrial Research and Medicine, SMRM, India, and the Department of Biotechnology and Biochemistry. In the month of September, special talk on career opportunities in life science after graduation was arranged for third year and second year biotechnology students. Model presentation on nature and energy conservation was arranged for third year and second year biotechnology students. In the month of September, and guest lecture on enzyme kinetics and its regulation was arranged for first year and second year biotechnology students. A guest lecture and association valedictory function was conducted and prizes were distributed for winners of various activities organized by the association. Students' achievements. Ms. V. Ritu of 3rd BSc Biotechnology completed summer research fellowship offered by Indian Academy of Sciences, Indian National Science Academy, the National Academy of Sciences with a fellowship amount of ₹25,000 under the guidance of Dr. Ganesh Chandra Sago, Rajendra Memorial Research Institute of Medical Science, Patna during June to July 2019. 5 of our students, Ms. K. Nandini, Ms. M. Nityashri, Ms. P. Sweta, Ms. C. Geeta, and Ms. Mithila Shrikant have successfully completed NPTEL CEC online certificate course in various streams. Participation by students. Our department students had actively participated in various international seminars, national conferences, entrepreneurship awareness camp, youth leadership summit, and workshops organized by different universities and institutions in and around Tamil Nadu. Active involvement was also found in online quiz competitions organized by Ministry of Defense, Government of India. Students of our department had also enthusiastically participated in various clubs and forums of our college. Participation by faculty members. Faculties of the department were participated in Indian conference organized by Kongunadu Arts and Science College and refresher program organized by Center for Education Beyond Curriculum at Christ University, Bangalore. and had also participated in various national level events conducted by different universities. Thank you.
Thank you, Sweka. Now, I'd like to call upon Dr. C. Deepa Ma'am, Head of the Department, Biotechnology, to introduce our Honorable Chief Guest.
Good afternoon, all. I am very happy to introduce the guest of today, Dr. Prashanth Kumar Kandalla, Staff Scientist and Senior Postdoc, Stem Cell and Macrophage Biology Lab, Technical University, Germany. He completed his UG and PG from Andhra Pradesh. He worked on skeletal muscle stem cells at Center for Cellular and Molecular Biology, CCMB, Hyderabad. He did his PhD in Marie Curie University, Paris, in human skeletal stem cells on muscular dystrophies and on different diseases. From 2011, he is in Germany as postdoctoral fellow working in hematopoietic stem cells. He also worked in stem cells transplantation and infectious disease. He has published many research articles in reputed journals like Journal of Experimental Medicine, Nature, and many more. In 2018, he moved to the Technical University, where he joined at Center of Regenerative Medicine. He focused on stem cell biology and macrophage biology. He is also a very good teacher and takes care of master's students. Apart from these, he is also an all-rounder who knows classical dance, including Bharatanatyam and Kuchipudi. He is a multifaceted person, and we are very happy to have Dr. Prashanth among us for this inaugural function. Dear Prashanth,
Hi.
you can take over the session. Thank you.
Okay. Thank you very much, Dr. Deepa. It was a very kind introduction about me. And good afternoon to all the students and others. And first of all, I would like to thank Thiru Palanisamy Avargal, the correspondent, and also the principal, Dr. Raman sir, for including me in this wonderful session. And also special thanks to Dr. Deepa to include me here. It's a great pleasure to be included in this. And it's wonderful to see all the evolvements and also everything like the work that are being done here, even though during COVID-19 situations, so not to be disappointed. Like all the staff are also continuing to motivate and also keep updated the students. It's really a great job. Thank you to one and all. Hopefully the total world will be out of this pandemic very soon. I pray to the Almighty God. So let's move on to the today's topic.
Dr. Prashanth, you can share your screen.
Yeah, I'm trying to do, yes. Do you see my screen?
Yes. We can see, but yeah. Yes.
Okay.
Yeah, full screen.
Yeah, so please let me know if I am fast or slow or whatever, and if my audio is not clear or anything, I can immediately adjust accordingly.
Okay, thank you. Yeah, definitely.
Yeah. So when Dr. Deepa approached and explained about the students, I thought about presenting something very basics about the immunological aspects, and the main thing will be like antigen presenting cells. This is the more basic it would be. So I thought this topic would make sense about this session. So today I will present about this topic. So before going into the topic, I would like to just say that throughout my research career, I was mainly focused on the stem cells. So what, and especially adult stem cells. So what is an adult stem cell? It can self-renew to maintain its own population, and also can differentiate into different cell types that are necessary in the body. And stem cells can be seen, like, for example, there are many kinds of cells, stem cells in the body, from the bone marrow fetal and the cord blood, and also in the tissues like muscle and every like connective tissue and all the other tissues. These stem cells are implicated in cellular therapies to encounter hematological malignancies and diseases of the immune system. They also participate in the regeneration and the repair, and also they are implicated during aging. So this fascinated me to work about this thing, and also to further focus about the differentiation of these stem cells into different cell types, and especially myeloid compartment, which is like, for you, if you know, it's macrophage kind compartment. So just to, okay, so just to tell you, so this is the, do you see my cursor, Deepa?
Yes, Prashanth, we are able to see your cursor.
Okay, thank you. So this is the hematopoietic stem cell, and then it is called hematopoietic tree in general. So what the stem cell first produces some progenitors, and these differentiate into further downstream progenitors, and finally you have all the immune cells. So basically you would have heard about macrophage, monocyte, red blood cell, and also the lymphocytes in your basic science in your previous classes. So all these cells are produced from the progenitors or upstream coming from the further progenitors and also from the stem cell. So this is the basic, in a classical model, this occurs. But when there is a need in the body, this stem cell, with the help of various signals in the body, can directly produce different mature cells also. So today we can focus on this compartment where the stem cell goes towards the macrophages. So this is our main focus of the lab. Of course, I'll not go into those details, but my focus will be on antigen presenting cells, but I'll just give you a brief introduction. Why is this compartment very important in the immune protection? Sorry, the slide jumps. So if you see, the stem cell can produce first monocytes, and these monocytes can become macrophages and reaches the tissue. So this tissue, like in one pathway, or these macrophages can also be derived from the embryo itself. When the embryo is forming, there are certain progenitors which produces macrophages, and then they are maintained throughout the life. So the macrophages, they are present in all over the body, and each macrophage is called differently. So the macrophages, for example, the macrophages residing in the bone marrow are called bone marrow derived macrophages, and in the liver they are called Kupffer cells, and in the lung they are alveolar macrophages. We can come to about alveolar macrophages at the end of the talk, maybe if someone is interested, but these macrophages can be derived either embryonically or from stem cells, or they can also self-maintain by proliferation. So, and if you see, the macrophages can aid in different functions such as tissue repair regeneration, cytotoxicity, tumor infiltration, phagocytosis, and immune surveillance, and also in the antigen presentation. So all these functions requires majorly the antigen presentation. Let's go for each example. So how does a macrophage goes in the tissue repair and regeneration? So for example, if you see, during the evolution from a smallest organism to the biggest organism, the regenerative capacity is going down, and the immune competition is also going down. Where if you compare to the fetus in the humans itself, the fetus will have high regenerative capacity, but the immune competence is low. But when at the adulthood, the immune competence is high, whereas the regenerative capacity is going down. So it's really interlinked. So it's very important to always think in this direction. When we think about immunity, it always depends on the age of a person also. So if you, for example, here, when we see about heart regeneration in day-to-day life, you are hearing about cardiac injuries or cardiac arrest or heart attack or anything. So what happens here? If there is an injury here, there are different methods of injuries, but here in the myocardial infarction, what happens? There is innate immune response, and the stem cells immediately come into action and they produce the necessary macrophages. And in the macrophages, for example, in the case of young adults like or neonatal kids, for example, when the macrophages form the new, it participates in angiogenesis and gives new cells, and then there is a complete regeneration. Whereas in the adults, what happens, these macrophages are not completely participating in this. There are other factors are involved, and the fibrosis occurs instead of angiogenesis, and this forms the scar here. So what happens when the scar is formed is there is no proper, sorry, the slide jumped. So there is no proper functioning of the heart. When there is a scar, it forms a very tough tissue, so the heartbeat doesn't go on and it ends up. So let us see another example of phagocytosis and immune surveillance. So what happens in the human body? In the steady state, the bone marrow from the stem cells, the steady state myelopoiesis and lymphopoiesis occurs. So production of myeloid cells is called myelopoiesis. Production of T cells, B cells and others is called lymphopoiesis. And when there is an infection, what happens during a bacterial or fungal or viral infections, it enters the bloodstream, and then the stem cell reacts to it, it gets activated, and then it produces more myeloid cells in the first defense, because these are in the innate immunity. So I'll come back to the topic soon, but just to mention here, there are more macrophages and which come back here to fight against this infection. So, and then in our lab, what we also was working was like we found that there is a cytokine called myeloid cytokine, which is low in the steady state and it gets increased in the MCSF. So what do we see in the daily life? Like for example, in the case of bone marrow transplantation, what happens that the patient is myeloablated. That means the host immune system is depleted and you give new cells to produce new immune system. So for example, the stem cells are given. So our idea is, as there is high MCSF seen during infection, what our idea was to give MCSF so that it can already boost this process. So imagine there are more macrophages before, after this, and when the infection occurs, it combats very easily and the patient is saved easily. So this part of the work has been published in Journal of Experimental Medicine, and now we are going for clinical trials in Germany and also in France and Switzerland in collaboration with different projects. So this is also, in this case, we found that macrophages play a major role. And then, so all these functions are mainly mediated by antigen presentation, especially the immune protection. So let's go into the details of antigen presenting cells. What are in general antigen presenting cells? So the main agenda of this talk would be like, I'll go through introducing some terms like innate immunity, adaptive immunity, and the different immune cells that are involved in this process, such as macrophages, dendritic cells, and natural killer cells, and the lymphocytes such as B and T cells. And then I will go on into further details, because this process includes the major histocompatibility complex, class 1 and class 2 molecules. So let's go into details stepwise. So what happens when there is an infection? So when there is an infection, the microbes, for example, let us say microbe instead of bacteria, fungus, and virus in specific, we can call them a microbe. So when there is an infection, they cross the epithelial barriers and then send the signals. So through the blood, all the inflammatory signals are coming to the main phagocytes, or the main things are called, so macrophages are the main police. For example, if let us sit back and think, if there is some crime in a city, what happens? Someone calls and says, hey, someone calls to the police and say, hey, something is wrong here, please come here. Immediately the police come into action to see the first, to solve the problem as soon as possible. So this is what the macrophage does. As soon as there is an infection, the first thing come into the action is macrophages. And then later on, this signals is sent to another cell type called dendritic cells, and this further sends the signals to downstream cells such as B cells and T cells. So this is like a kind of, if you can imagine in a scenario of something crime happens and how it is solved. So, or also at the national level, if you think about a like at the army level, it goes the downstream like that. So the first army is like macrophages. So these are the macrophages coming into action, and also they signal further another cells such as called natural killer cells, because this can directly also engulf or kill the pathogen. So this is called innate immunity. So in the innate immunity, the main actors are macrophages, dendritic cells, and natural killer cells. Let's then come to adaptive immunity. What is meant by adaptive? The name itself says it is adapted. That means as soon as there are signals that gets by macrophages, dendritic cells, and natural killer cells, and when they can't finish the job themselves, what they do is they send the signals to other cell types such as B lymphocytes and T lymphocytes, and that's where they come into the action. So they are adapted to do some more functions. So that's why it's called adaptive immunity. So these are the two main cells that are involved in adaptive immunity. So let's go into details in one by one. So first we can focus on B lymphocytes. So what does a B lymphocyte do? So when there is a microbe, so it is recognized by the B cell and it gets activated and then produces a lot of cells called plasma cells, which in turn produces the antibodies. And in turn, this antibody again combats onto the this pathogen, and it further processes and takes it to T cells and then natural killer cells to kill it. So in this process, there are two things happening. So one is so in this you have to remember an epitope is a very important determinant called, it is antigenic determinant. Antigen is nothing that the protein or peptide that is present by the pathogen or microbe. So the antigenic determinant is present on the pathogen. It is specifically recognized by the antigen binding side of the antigen receptor of, for example, B cell or T cell. It has a receptor called B cell receptor or T cell receptor, or in the case of B cells, an antibody. So this antigen can be two kinds. For example, it's called conformational determinant, where it has a specific conformation, and this binds to the receptor or antibody, and then this is denatured, and then this denatured is normally recognized by the further cell type. I will come back to that later. In the linear case of determinant, it has like two kinds of determinants. One could be accessible, one could be inaccessible, because when the protein folds, this can be hidden. This determinant can be hid inside. So what happens in this case is the receptor recognizes the accessible determinant first, and then in one way it denatures to expose the inaccessible determinant, or in the other sense, it can also further denature this protein completely. So that's making the pathogen or microbe inactive for its pathogenicity, for example. So, and let's go back to the T lymphocytes. How does this go? So in the case of B lymphocytes, it goes through the plasma cells and produces antibodies and fight against the infection. Whereas the T cells, they do it in a different way. So the T cells, they always bind either to B cell or other cell types such as macrophage or dendritic cell, and then further it becomes effector T cell, and then further downstream it goes into the action. So let's go more into detail. So what happens? So in contrast to B cell that recognize free antigen, T cells can't recognize a free antigen. It needs to be presented, this antigen to be presented by some molecules called MHC, that's called major histocompatibility molecules, which are present on the antigen presenting cells. So the main antigen presenting cells are macrophages, dendritic cells, and B cells that present to the T cells. So When there is only T cell and there is antigen, there is no T cell response. But when there is T cell, there is an antigen, and then the antigen presenting cell, this peptide epitope of antigen is presented by APC, is recognized by the T cell, and then this T cell response is present. Let's focus on this part mainly. So what happens and then it's also not every peptide can be important in this presentation. For example, when for example, this is a pathogen peptide or from the microbe coming, what happens? A macrophage engulfs it by endocytosis. It forms a phagosome and then completely breaks down all the protein, like all the infective parts of all the pathogens. Then it gives certain various types of peptides. So when this kind of peptide comes here, the T cell response won't occur. But when an active or immunodominant epitope is shown by this MHC class, this green colored thing is called MHC molecule. So on the macrophage, when this MHC molecule presents this epitope to the T cell, then it's get activated. So in this case, you have already seen it's specifically the T cell, which is expressing a glycoprotein, which is called complex of differentiation 4+. So in immunology, this is a very complex thing, but each cell type, you can recognize them with specific markers which are expressed on their surface of their cells. And this can also be isolated by a technique called flow cytometry. Maybe in further classes, your staff will explain how this technique is done, or maybe you have got introduction already. But every immune cell can be isolated recognizing these markers. So what happens, like when the epitope is presented, the CD4 cell get activated and it proliferates to combat the infection. So let us see more in detail. So if there is no antigen, there is no antigen peptide response. And not only antigen is needed, it also needs co-stimulation. So here, for example, there is a foreign body which is recognized by MHC class molecule, and on the T cell, there is a T cell receptor. But this is not enough to activate the T cell response. So what is needed for a T cell response is both antigen and a co-stimulation. For example, here the pathogen is broken down into peptides, and this is presented by the MHC class molecule to the T cell receptor. And parallelly, there is a connection which is called co-stimulation occurs. For example, the CD28 on T cell is recognized by CD80 or CD86, which is a marker like M2 marker, which can present macrophage marker. So for example, and it can further cross present, and then there is a T cell activation. So this is so it needs the first signal, which is the binding of this complex, and then the co-stimulation, which is necessary, this one, the second. Then the activated T cell really functions on the pathogen and it kills the pathogen. So let's go to the major histocompatibility complex. What are these? So these are the human leukocyte antigen. Maybe you would have come across in the case of bone marrow transplantation, people would be talking about, do you get a donor, HLA matched donor? So this is what? Human leukocyte antigen. So when the HLA, like earlier days, you have to give only, like for example, in the autologous transplantation, you have to give from the same patient to the same patient cells. Whereas nowadays, the advances are more in the allogenic transplantation models, where you can give, if the HLA is matched on the both, like the patient and the donor, you can use. So this you would have come across. So there are two kinds of molecules, class 1 and class 2 molecules. And the class 1 molecules are especially recognized by T cells which express CD8. And class 2 molecules are expressed by CD8 T cells, for example, lymphocytes in CD8. So in this way, you can also divide MHC class 1 and class 2. At the same time, these pathways can be divided into CD8 T plus T cell response or CD4 T cell response. And then this one is mainly expressed on all nucleated cells, whereas this molecule is expressed only on mononucleated phagocytes, such as monocytes, dendritic cells, B cells, endothelium, and thymic epithelium. So that's why this is the major response that occurs when there is a very dangerous infection or when there is severe infection. This is a kind of response occurring when there is a particular kind of pathogen is infecting the body. So let's go further. So the three major types of MHC class 2 plus antigen presenting cells for T cell activation or function, let us see one by one how they function. For example, let us take, this is the dendritic cell. The dendritic cell recognizes the pathogen and then it presents on the MHC, like once it is internalized in the cytosol, it breaks down the peptides and through the MHC class molecule, it presents the epitope. And this is recognized by the naive T cell, and this naive T cell divides and becomes effector T cells, and these proliferate in function. And what happens, the macrophages in the parallelly also can recognize this pathogen. It do endocytosis and breaks down all the peptides, and it increases its MHC class 2, and then it can present more robustly the epitopes onto especially the effector T cells, which were already activated. They recognize this cross presentation, and then they further kill all the microbe because of this reaction. So what happens in the case of B cell? This is a third MHC. So the MHC class 2 is present as soon as it recognizes the pathogen, and then it turns it with the help of effector T cells, it activates. Then effector T cells send some signals such as cytokines, for example. These secreted factors further activates the B cell to become a plasma cell. And this plasma cell will produce the antibodies which are streaming in your blood. So these are the three kinds of MHC class 2 pathways that is occurring in this kind of infection. So let us see, if you see on the B cells, they are highly efficient antigen presentation due to specific capture of antigens via the B cell receptor. So this is what happens. So when the macrophages sees the extracellular antigen, the antigen will be endocytosed and then presented on this CD4 T helper lymphocytes. Then this further produces the cytokines, and then macrophage activation occurs, destruction of phagocytosed antigen. This is one way. In the other way, the B cell takes this extracellular antigen, then the B cell, after internalization, goes to the T cell, which is the T helper cell in this case, and then this also produces the cytokines, and then the B cell produces the plasma cells, which gives antibodies, and then this B cell antibody secretion will kill the antibody, the bind to the antigen, and then it will make inactivate. So in one way, it can internalize and kill, and in the other way, the antibodies can bind to the peptides and block its effector functions of these peptides which are produced by the pathogens. So that is what you have seen on the, So this is where you have seen on the MHC class 2 presentation, and in overview MHC class 1, what happens, there is a cytosolic antigen. There is no endocytosis or anything. The antigen directly enters the cell membrane and is present in the cytoplasm of the cell. And this one is recognized. And then what happens, MHC class 1 molecule is coming up, like expressed more on the cell membrane. And this will present the epitope. And then here, in this case, the cytotoxic T lymphocyte, which is CD8 positive T lymphocyte, recognizes this epitope and then further kills the antigen expressing target cell. So in this case, for example, if there is a macrophage, the CD8 cytotoxic T lymphocytes even kill the macrophages. That's why when there are only, let's say there is a 100 macrophages are coming into the first defense, they are killed in this method. So what happens? The body loses the macrophages. So then further, the body recognizes the signals are sent to dendritic cells, which can further go through the bloodstream and reaches the lymph nodes and then activates the B cells and T cells. So these are the two MHC class 1 and class 2 pathways that occur. So this is MHC class 1 structure. If you want to know in detail, of course, this is more detail, but just to make you curious, I wanted to show like how they look. So each molecule, they have different peptide chains. So they have different substructures. And for MHC class 2, the substructure is like this. So if you want to see further, in the class 1, a peptide of 9 to 11 amino acids can fit. And in the class 2, 12 to 24 amino acids can be presented to, like as an epitope, can be presented to the T cells. So this is what as in an overview, you can see there is a T cell receptor, and this is the MHC class molecule. So we are going deep by deep from a cellular level to the molecular level and at the MHC class level. So stepwise at MHC class 2 looks like this. So there is a first contact made is by T cell contact residue of peptide. So this is the main peptide that is presented, and you can see there is at each level, there is a kind of lock and key kind of mechanism happening, and there is a recognition particular. So that's why one particular kind of MHC class can be recognized by particular receptors of T cells, because there are hundreds of receptors, they are exposed on T cells, and then there is a specific signal coming from specific molecule that is recognized. So that's why there is a specific action going on. So what is the physiological significance of these MHC associated antigen presentation? Let's go into the little bit more detail. So there are two kinds of presentation. One is extracellular and another is cytosolic antigens, like just use saw to different T cell subsets. For example, in the case of class 1, it is cytosolic, and then it aids to the killing of pathogen. And in class 2, like these ones, just now I gave all the details in above slides, and I'm just summarizing here. So the MHC class 1 molecule associated presentation occurs through cytosolic agent, and it culminates in killing of antigen expressing target cell. And in the second case, which is class 2 molecule associated presentation, the extracellular antigen is presented to the T helper cell, which produces the cytokines, and the killing occurs in two different ways, either by internalization and through phagosome and then killing by phagosomal activity, or the antibodies binding to it and making it inactive. So what are the cell biological mechanisms that can occur in these two, cytosolic and extracellular antigens? It all depends. So it is yet to be studied in various diseases. For example, even in the case of COVID-19, people are trying to understand what happens in that kind of situation, what are the mechanisms that are involved. Once we understand the mechanism, it is very easy to find a vaccine or to find anything against it. So let's talk that later. So just to see separation of two major intracellular compartments by membrane. So this, if you imagine this is a cell, so this is the nucleus, and nucleus has a nuclear membrane, and there is one more nuclear membrane body, which is called Golgi apparatus. So for example, when there is a secretory vesicle is there, it endos, like for example, okay, let's see first here. So when there is a pathogen coming in this, it goes into endosome, it forms endosome, and then it forms a phagosome, and then which releases lysosome enzyme, which makes the environment acidic so that the peptides are broken down. So then it's called, in particular case of macrophage, it is called phagolysosome here. And then further it is presented and killed. This is one way. Whereas in the secretory vesicle method, it happens, what happens like in the Golgi apparatus, it processes the peptides that it takes, then it secretes through the secretory vesicle, it secretes all the peptides that could be presented for further downstream. So these are the two major things that are important is cytosol, which is this one, and vesicular system, this one. So the pathogens are very clever. Either they can infect the cytosol or through the membranes. So let us see what are the cytosolic pathogens that do. So the cytogenic pathogens, they always occupy or attack the cell plasma, the space, and then they eat up all the things that are present. So that's how they can kill. But these, when presented, these are mainly presented by MHC class 1, like you saw earlier, through the CD8 T cells, and then it occurs when they are attacking the cytosol, the total cell. For example, in this case, the macrophage leads to cell death. Whereas when the presentation is through intravesicular pathogens, for example, in the vesicles, for example, in these small pockets, if the like let's see an example of macrophage here. So it goes through the endocytic vesicles, and mainly MHC class 2 is activated, and then CD4 T helper cells and effector cells are involved, and then they become activated to kill intravesicular bacteria and parasites. And in the third case, extracellular pathogens and then toxins, for example, they produce certain molecules which are recognized by the B cells. In this case, for example, they get internalized, and this is processed, and then they are presented through MHC class 2 molecules, and then these are recognized by CD4 T plus cells, and then activation of B cells to secrete immunoglobulin to eliminate extracellular bacteria or toxins occurs. So these are the mechanisms that going through. So let us see the overview. So very simple, the antigen presentation occurs by 3 different cells mainly. They can be from innate or adaptive immune system. From the innate immune system, macrophages and dendritic cells are the ones which present with MHC class 2 molecules. And then from the adaptive system, B cells are the ones which present the MHC class 2 molecules, and they have these are the 3 ones, they are called antigen presenting cells. They present the antigens or the epitopes from the pathogen or microbe to the effector cells, such as called lymphocytes, T lymphocytes. And if this is occurring through when the peptide occupies the cytosolic protein, the peptides in cytosol is broken by proteasome, and this one is first the MHC class 2 is internalized, and when the epitope binds here, it is shown by like, so first the peptides in the cytosol, they enter through the like here I forgot to mention you, it's more details, it's not necessary, but still I just wanted to know how you can ask how come a cytosolic protein can enter the endoplasmic reticulum. So here there are certain proteins called tap proteins. They will be closed in the beginning, but these peptides, they are very when they recognize certain bonding, they enter through these tap proteins and enter the endoplasmic reticulum to occupy and kill all the cell. So what happens initially, the endoplasmic reticulum, it has MHC class 1 internalized, and it is produced here. Once this is there is a perfect match for this, what happens, the endoplasmic reticulum in another way, it sends out into the vesicles, and then this vesicles is expressed on the cell membrane, and then the MHC class 1 is presenting the epitope to the CD8 plus T lymphocytes. So this is called MHC class 1 pathway. And in the next case, when there is a extracellular protein, they are internalized, they form an endosome, and then the endosome, like the cell produces also the lysosome, which enters lysosome enzyme, which enters this endosome, and then further it makes the pH acidic, and then this will break down this peptide into small epitopes or small peptides. Then this one combines to phagosome and it forms phagolysosome. Then further what happens, meanwhile parallelly the endoplasmic reticulum is preparing the MHC class 2 with an invariant chain, and this one is it sends out into a kind of phagosome, and then it fuses to form a phagolysosome. So where this particular MHC class 2 is when there is a matching for the dominant or dominant peptide, it recognizes, it binds to it, then this one is presented on the cell membrane, and then the MHC class 2 molecule presents the epitope to the CD4 plus T cells. So this is called class 2 MHC pathway. So in the class 1 MHC pathway, the main thing you have to remember is the pathogen is cytosolic, and then MHC class 1 molecules are present on the membrane, and T lymphocytes such as CD8 positive cells will come into action. In the class 2 pathway, there's extracellular protein, MHC class 2, and then CD4 plus T lymphocytes. So this is very easy to understand like that. So anyway, I have just made a chart for you. Maybe I can share this later, comparing the MHC class 1 pathway and MHC class 2 pathway, how the different features vary in these cells. So I can share these things with Dr. Deepa later, and she can share with you all. So this is our lab, very fun-loving lab all the time. And I'm sitting here, think like the concept of this box is like, we say it's like think out of the box. So you have to, you are in the box always, but you have to think out of the box. So thank you.
Thank you, Prashanth. It was a very nice presentation explaining all the basic details about immunology. And I hope all our students would have enjoyed your session. And now, dear students, the session is open for discussion. And if you want to ask any questions, you can put it in the chat box. You can type your questions in the chat box so that it will be presented to the guest, for which you will get the answers.
Hope I didn't bore anyone.
No, definitely.
Our students didn't run away.
They are still here. They are still here.
Feel free to interact. Don't hesitate. Anyone can also write their questions or anything related. Or even if you didn't understand some particular part, whatever it is.
So, Prashanth, I just want to ask one question.
Yeah.
How many days will they take for that HLA matching? Like you were telling about when during bone marrow transplantation, they go for HLA matching. So
yeah.
Usually, what is the procedure like? How many days they'll take, and
it's few hours now. Nowadays, there are so many ELISA kits. They can do it very quickly within few hours here. Even in India, it's well advanced, and the doctors are much more advanced, I would say, in India, because they are very famous for this. That's why many doctors, they are hired in US also. So this matching is really done very quick, actually. But the only problem is the test is very faster, but the getting the right person, right donor is very rare. That's why it's very difficult. For example, when people go for organ transplantation also, this is very important that the matching of the HLA is very important. HLA or MHC is very important between these two patients.
Okay. So
but normally it's done in within few, one day normally.
Okay. Okay. And we have another question from one of our students, Ritu. Like she wants to know, like MHC class 1 and MHC class 2, which one is evolved first?
Okay, so For example, you saw that MHC class 1 is specific. It's not about which one first or which one second. If you want to ask between like innate and adaptive immunity, the innate immunity is first and the adaptive immunity is second. In the case of MHC class 1 and class 2, the main difference is what kind of pathogen or what kind of microbe is present. If a microbe is cytosolic, that means a microbe can enter a macrophage or dendritic cell through the membrane and can sit inside the cytoplasm and can enjoy like a kind of it's like it can home there, and then it feels like, okay, I have reached here, so I can attack this cell. So if a peptide or pathogen goes into cytosol, this leads to MHC class 1 pathway. But if there is a pathogen which is not faster or robust, for example, because you also have to think in a way, for example, when you are, that's why people say cleanliness is more important, all these things. So there are two things. So if you have 100 molecules of bacteria, maybe the MHC class 2 is faster because the first macrophages come into action. They eat bacteria, kill the bacteria very fast. There are like, say, 1 million bacteria attacking your body. So in this case, what happens? The available macrophages, first it come into action. They can kill all whatever is possible, like 100 or 200 or 1000, for example. What happens to the rest of the bacteria, which is still is aggressive? Then the macrophages or dendritic cells, if they are not able to do their job, the innate immune cells, if they can't do their job immediately, they send signals to the adaptive cells. So this is a kind of stepwise process. So what comes first is all depends on the pathogen.
Thank you, Prashanth. And students, do you have any more queries? Even if it is going to be a basic question, it's okay.
Yeah, please don't worry. You can ask any kind of question. Don't worry. You can. It's always good to interact, because when you start interacting, you learn more. You should remove your inhibitions. Then only you will learn more. So the enthusiasm will be more. So feel free to ask. Don't think someone will laugh at you, someone will smile at you. No, don't think about anyone else. Just think about you. You want to learn something, just go ahead and attack the person. That's it. Attack with your questions. Beat any teacher.
So we have another question. I think they are motivated by your words. And the question is, when do gut bacteria change into a pathogen?
When do gut bacteria change into a pathogen? Okay, it's a very good question. So normally we know there is a good bacteria and bad bacteria, right? So what are good bacteria? Let's give one example. For example, lactobacillus, which comes from your milk. So the lactobacillus is called good bacteria, but because it can produce certain enzymes which can break down certain proteins that is helpful for our body, and it can assimilate, and it is useful for our development. There is a bad bacteria, for example, a Shigella or some other very dangerous pathogens, like may not be bacteria only. Let us say Aspergillus, a fungus. So Aspergillus, where do you see Aspergillus? Can anyone answer? You see this every day in your life, Aspergillus fungus. Okay, I will tell you. In your house, you have kept some food and you forgot. Okay, someone forgot the food somewhere in your corner. Maybe you made a vada, for example, or some bread, and you forgot on a table or in some corner. Next day or day after tomorrow, if you realize and see there is a green thing growing on it. Did you come across that? Most of it is called Aspergillus species, so the fungus. So what happens sometimes, this you can see when it is green and growing like a kind of big lump, you can easily see this green thing, and you can immediately say, okay, it's gone, it's rotten. So you will throw into the dustbin. Imagine there is only 10 spores on the food. So you were talking to someone, So your amma calls something and you said, do this job first. So you leave the plate or go somewhere. So accidentally some air passes and then there are some fungal spores sitting on it. What happens? So this you will eat already. So this is also a kind of bacteria or fungus that you are going into your gut, for example. So this one, when it goes in lesser quantity, it's fine. But imagine if you take it inside and by chance you were already not so immunely healthy or something. What happens? This pathogen, after entering your body, they increase in your gut. They multiply, and then they become, be it a bacteria, fungus, or virus, whatever. They increase and then they take over the gut. So this is bad bacteria. And certain bacteria, such as E. coli, for example, Escherichia coli, there it can act good or bad. For example, if they are in a certain amount in your gut, it always helps for the assimilation of your digested products or whatever it is in your small and large intestine. But when Escherichia coli increases in numbers and becomes too much aggressive, it can be very dangerous for you. It can lead to diarrhea and some other problems. So there is, that's why you can say that the gut has certain capacities to keep the good bacteria, but if you replace it with some other bad bacteria, then it can be harmful for you. So there is always a kind of balance that you have to maintain. That's why we are always asked to do like healthy habits, to keep healthy, to run, to exercise, so that your body maintains certain good bacteria in you and certain physiological things in a normal way. So eat healthy, like drink healthy. For example, they will not ask, don't drink anything anywhere. So these are the reasons that they say. So until certain level, the gut bacteria is always good. But when you replace it with certain pathogens which are coming from outside, then even your good bacteria can't help you. It can go in a negative way.
Okay. So we have another question. What happens when phagocytosis fails?
What happens when phagocytosis
fails?
Okay, so it's also another good question. So that's what I showed you in one of the slides. So phagocytosis, when does this first occur? In the innate immune response. So when there is a pathogen, the macrophage come into action, it engulfs the pathogen, and then it forms through endocytosis, it forms phagosome, lysosome, phagolysosome, and then it kills the bacteria. So this process is called phagocytosis. So what happens when this fails? That's what I already explained a bit. So when the macrophage can't phagocytose these cells, it sends some certain signals to other cells, such as natural killer cells or dendritic cells. It sends it secretes certain molecules or certain proteins, let's say cytokines. They are called cytokines. So they send some certain cytokines to activate dendritic cells. For example, macrophages or monocytes, if they can't phagocyte enough, what they produce, they can produce interferon gamma, which is also a secreted molecule. The interferon gamma then goes to natural killer cells, and it says, hey, the macrophage is not able to do its job. So okay, do your job. So that's what the interferon goes to the natural killer cell, and then a natural killer cell gets activated, and it does its job in a different way. It goes to the region of infection. It sees, oh, okay, there are so many pathogens here, I have to take care of them. So they will go and kill all the kill the virus or other aggressive fungus, for example. In most of the bacterial or fungal cases, the macrophages are more effective already. But in certain viral cases, when they are aggressive, the macrophages can't do their job enough. So in this case, the natural killer cells will also come into action. And the natural killer cells, the name itself explains, they're naturally killing. So their job is to kill. So they can kill all kinds of viruses mostly. And even imagine certain cases, they also don't have enough natural killer cells. That's where the body reacts in a different way, and the patient dies due to sepsis, means different kinds of infections. So in the case of sepsis, this is what happening. Whatever the question you ask, this is seen in the case of sepsis. Sepsis is a kind of clinical condition where the patient dies because of several infections. So that's what happens in this case. Very good question.
So how phagocytosis happens in case of autoimmune disorders?
Okay, so this is the very critical thing in one of our institute. In our institute, one of the lab is also working on this, such as autosomal diseases, for example. Here, for example, in the case of phagocytosis, it goes in a negative way. The macrophages or something in the transplantation studies, this is what you see. When the cell of our immune system doesn't recognize the foreign body, then it will attack. But when it recognizes, then it does its job in the right way. So this is what happens in the autosomal diseases. It will be unable to recognize its own thing, and if it is foreign. So that's why these people with autosomal disorders, they are affected in an adverse way if you enhance the phagocytosis, for example.
Okay, so actually one more person has asked a question. I think you have your answer is there already. Like when few people have autoimmune response, whether they have higher immune response. So I think you have already answered for that also. So one more continuation of the previous, like when do the gut bacteria becomes pathogen? So they have asked so it can be only replaced, like pathogens can only be replaced. It cannot destroy itself like autoimmune disease.
Your voice was breaking. Can you please repeat? Sorry.
Hello, can you hear me?
Yeah, now I can hear you.
Okay, like it is the continuation question for the student who asked whether gut bacteria can be converted into a pathogenic bacteria. So now she is asking, so it can only be replaced, so it cannot be destroyed itself like autoimmune disease.
Mm-hmm. Yes, that's correct.
Okay.
Someone also asked whether secondary infection are fatal.
Yes.
No, there is one more before. Can we control the regeneration of the cells and also the production of macrophages? That's a good question, and for sure we can control this based on now. That's what the treatments are focusing on. So, for example, in the clinics, when people are particularly, let's talk about the muscular dystrophies. In these patients, what happens? The muscle, like there is no normal development. For example, the cells that are forming the muscle is called myoblast. So when they proliferate and then fuse to form myofiber, and many myofibers form to form bundles called muscle fibers, and different muscle fibers form muscle. So this is normal in a healthy person. But in the patient of muscular dystrophies, this is not happened, and there is the muscle is very weak. And in certain muscular dystrophies, even the child dies within 5 years. So in this case, what happens is people are trying to improve the condition of the child by various methods. So you can transplant, you can do cell therapies or gene therapy, for example. So when you do, or genetic modification of certain things in the body. So what happens in the cell therapy? You transplant cells, and then the regeneration of the cells occurs there, and then particularly you can improve this. So this is a case of muscle. For your question was related to production of macrophages. So that's where we do in the bone marrow transplantation. For example, to treat cancers, what happens? Like there is a patient with cancer coming to the hospital. So what the doctors do, they first do irradiation or chemotherapy to deplete his own immune system. Then they find the right donor, and then they transplant these stem cells, or bone marrow stem cells, it's called. These stem cells, once they are transplanted, that's what they regenerate to form different cells. And imagine the hospitals, even though you feel they are super clean, but not really. In the many kinds of infections occur even after transplantation of cells, even though the transplantation is successful in a patient, but the patients might die due to different infections, which is also termed as sepsis. So once the patient is transplanted with stem cells, what happens in the hospital? The stem cell, it takes time. For example, it takes 30 days to produce macrophages, 40 days to produce B cells and dendritic cells and other cells. And then within 30-40 days, and natural killer cells are also produced from the donor-derived stem cells. And then it takes 60 to 80 days to produce T lymphocytes. So that's why different kinds of infection can kill a patient at different times. Imagine there is a bacterial infection within 30 days to 40 days. What happens? Already the stem cells are producing these macrophages. They do their job. Imagine there is a viral infection. So there is not enough for a macrophage to do the job. It needs natural killer cells. So that's why the natural killer cells are also coming into action. Imagine this is also not sufficient. They also need T lymphocytes to do the job. So what happens at the 30 to 50 days if the severe viral infection occurs? This patient dies because there are no T lymphocytes, because the kinetics of differentiation or forming cells from the stem cells are different. So that's why nowadays, in certain cases, people are even trying to transplant macrophages directly. And even they are manipulating the macrophages by gene editing. They can also particularly express certain genes to function in certain tissue or in certain body part. I know it's a very wide topic, but hope I could answer you. Then I'll go back to the other question, whether secondary infection are fatal or dreadful. It could be. That's the reason. It all depends. For example, the primary infection is there. You have developed your immunity. All your immune cells have done their job. The B cells also produced antibodies. That's the concept of vaccine. So the vaccine, what you do is you take out the bacteria or whatever, you kill it, then you produce the antibodies. So this killed bacteria or whatever pathogen, that is given as vaccine so that your antibodies are enhanced, right? So this is one example. So in the secondary infection, what happens if you were healthy enough, there is a memory. The first infection, whatever has occurred, the stem cells and certain cells, like even in T lymphocytes, there are memory cells. Even in B cells, there are memory cells. Even in macrophages, there are memory cells. Even in stem cells, there is a memory cells. So what happens, you know the nucleus. In the nucleus, inside the chromosome, there is a chromatin. It's a kind of, what do you say? It's a kind of brain for it, okay? For the cell, it's a kind of brain. So everything happened before, it remembers in certain way or certain way. So when the secondary infection occurs, immediately the cell gets activated, and then it can come back very easily because it has a memory already. In the case of non-immune people, that means immune deprived people, this is lacking, and that's where the secondary infection could be fatal or dreadful for them. That's why we are taking vaccine, so that the known pathogens don't attack us in our whole life, like BCG and all the polio vaccine, everything you take. That's why you are never getting attacked by these pathogens in your whole life later, right? Yes. Okay, and then what causes immune system to overreact? So this is what it happens in the autosomal diseases. The immune system, when it doesn't recognize self or foreign or whatever, it overreacts to the body. So there could be certain, your question is what causes? This means there can be some genetic defect in that person. It could be genetical, or it need not be genetical. Sometimes during the evolution or during the development of a patient or a person, you may get certain acquired diseases which can also affect your immune system. So in this case, in certain cases, the immune system can underreact, or in certain cases, the immune system can overreact. So when it is underreacting, it becomes fatal, or if you can't fight the infection. If the immune system is overreacting, that's where you get autosomal diseases. So that's where that's another condition. It could be, for example, in the case of MHC class 1 and MHC class 2, there are also syndromes which are particularly if they are lacking this MHC class 1, there is one a particular syndrome, but there is no treatment for this until now. But for MHC class 2, at the moment, people are doing stem cell transplantation and they are correcting this. For example, all the cells lack MHC class 2. That means this patient is genetically not expressing this molecule. In this case, we give stem cells so that these stem cells can produce more cells which has MHC class 2. So they do the job.
Thank you, Prashanth. I think students are clear with their doubts, and we are very happy that they came out with such a lot of questions, and they were able to interact very nicely. So thank you for all your answers, patient answering. And
it's my pleasure. Thank you very much, Deepa.
I call Sai Shweta to continue.
I'd like to invite Ms. L. Kirbashni of 2nd BSc Biotechnology to propose the vote of thanks.
Good afternoon, one and all. I would say that thanks are the highest form of thought, and that gratitude is doubled by wonder. Thank you is the best prayer that anyone could say. And on that note, I would like to thank our beloved correspondent, Thiru K. Palanisamy Avargal, for his constant endeavors on our growth. I would like to dedicate my sincere thanks to our principal, Dr. N. Raman sir, for lifting us up and up through his experiences. To be inspired is great, but to be an inspiration is honor. On that way, I thank our chief guest on behalf of our institution, Dr. Prashanth Kumar Kandalla sir, staff scientist and senior postdoctoral, Stem Cells and Macrophage Biology Lab, Center for Regenerative Therapies Dresden, Technical University Dresden at Germany. Thank you, sir, for inspiring, encouraging, and supporting us. My heartfelt gratitude to our pillar of support and the head of the department, Dr. C. Deepa ma'am, for her tireless effort. A delightful thanks to our faculty members for guiding us towards the wellness. I extend my thank note to the heads of various departments, faculty members, and non-teaching staffs for their guidance on our growth. Students too hold the gratitude for their cooperation. The way a team plays as a whole determines its success. Finally, I thank the organizing team for this successful event. It's my privilege to propose the votes of thanks on this interactive occasion. Thank you.
Excuse me, pardon me for intruding. I would like to share something quickly. So I would like to say a very special thanks to Dr. Deepa. You have a very nice and motivating person in your institute, I would say, because we both work together in CCMB. I know since 2003 now. So since 2003, I know Deepa very well. And then she has been always a very dynamic person and very motivating person, and always very cheerful. And then it's very nice to have around such a person, and very enthusiastic also. And I wish her all the best, like throughout her career, that she does this kind of great jobs, and then encouraging all the students. And then, yeah, please also students, please don't miss the opportunity. You can gain a lot of knowledge also from Dr. Deepa. She is such a nice person. You can always approach her in very nice ways, and she's there to help you. It's wonderful to have her. Thank you.
Thank you, Prashanth. Thank you for your words. And really, our department is very thankful to you for your nice presentation and your nice words. Thanks a lot. And we hope we'll again meet you with another nice topic, which will be very interesting to our students.
Oh,
So,
that's wonderful. Thank you so much.
thank you. And I thank our correspondent sir, principal sir, and PD sir for providing the Zoom link and giving us the technical support during this time. Thank you, PD sir. And thank all the all the students who were patient listeners for this function. Thank you all, and thank you once again.
Thank you one and all once again for your precious time and marked your presence here. Thank you.
Thank you. Bye.
Thank you, Prashanth.
Be safe.
Madam, wind up பண்ணிக்கலாமா?
Everyone.
Yes, sir. Yes, sir.
Okay, madam. Okay, thank you.
Okay, thank you, sir.